The 2026 ACC/AHA Dyslipidemia Guidelines recommend that every adult in the United States get their lipoprotein measured. Every adult. Once in a lifetime. This is a class of recommendation 1. That's 260 million people. Now let me tell you what the guideline recommends that you do when you find it's elevated. You optimize modifiable cardiovascular risk factors. That's it. That's the treatment. The same thing you should already be doing for every one of your patients you've seen, regardless of their LPA. We're screening 260 million people to tell them something we would have told them anyway. That deserves a closer look. So what is LPA? Well, lipoprotein A is an LDL-like particle with an extra protein attached called alpha lipoprotein A, coded by the LPA gene. It's atherogenic, pro-inflammatory, and thrombogenic. It's also almost entirely genetically determined. Lifestyle and diet don't move it at all, and statins barely move it. Approximately 20 to 25% of the world's population has elevated levels, depending on the threshold you use. And that threshold itself is a problem we'll come back to. To understand why the guideline made this move, you need to understand the evidence. And the evidence is actually good on one specific question. LPA is a causal risk factor, not just an association, but causal. The strongest observational data set is the Women's Health Study, which was published again in the New England Journal of Medicine in December of 2024. Over 27,000 healthy women were followed for 30 years. Over that period, 3,662 first major cardiovascular events occurred. The adjusted hazard ratio comparing the top and the bottom quintile of LPA was 1.33, with a confidence interval of 1.21 to 1.47. That's a real signal. Elevated LPA, specifically above 44 milligrams per deciliter, tracks with meaningfully higher 30-year cardiovascular risk. Two things worth noting. First, LPA was elevated primarily in the top quintile. It was not the smooth dose-response gradient you see with LDL. The risk lives at the highest end. Second, unlike LDL, the predictive value of LPA did not attenuate over time. It kept predicting risk all the way out to 30 years, and that's something you could expect from something that's genetically fixed. If you're comparing, LDL had a hazard ratio of 1.36, and CRP, which is inflammation, had a hazard ratio of 1.7. There were a couple of other limitations to flag. First, 94% of the cohort was Caucasian. LPA levels and risk vary significantly by ancestry. Levels are highest in individuals of African ancestry. This cohort underrepresents that risk. Here's what the 2026 guideline can offer you when you find an elevated LPA. Optimize modifiable cardiovascular risk factors. That's the COR1, strongly recommended. Treat LDL aggressively, control blood pressure, don't smoke, improve lifestyle, good advice, but not LPA-specific advice. For patients with established ASCVD and elevated LPA who haven't reached LDL targets, the guideline recommends adding PCSK9 inhibitors. The PCSK9 inhibitors reduce LPA by about 20 to 27%. That's not what they were tested for, and the benefit in those trials was driven by LDL outcomes data, not LPA. We do not have a complete randomized trial showing that specifically targeting and lowering LPA reduces cardiovascular events. Not yet, anyway. The first cardiovascular outcomes trial specifically designed to answer that question is the LPA Horizon trial, which has over 8,000 patients with ASCVD and elevated LPA. Patients are randomized to pelacarsen, a new drug targeting LPA reduction by 80% versus placebo. As of the filming of this video, those results have not been published, and the guidelines we're talking about today just told you to screen every adult without those results. Let me ask you the uncomfortable question directly. When you screen 260 million people for an inherited risk marker that you currently cannot treat specifically, what are you doing? Well, one honest answer is brilliant proactive medicine. You identify high-risk patients now, counsel them aggressively on everything else, and have them ready to start targeted treatment once those therapies are available. That seems like a legitimate clinical rationale. But here's another honest answer. You are priming the largest possible market for an incoming class of drugs. A nation of patients who know their LPA is elevated, who have been told by their doctor that they have an inherited cardiovascular risk factor with no treatment. These will be the first folks in line when any of these new drugs get approved and costs whatever injectable cardiovascular drugs cost in 2027. Those two answers are not mutually exclusive. Medicine and pharmaceutical market development have always coexisted in cardiology. The question is not if it's happening. It's whether you, the clinician, know which part of the story you're in when you order the test. And then there's the assay problem. LPA can be measured in milligrams per deciliter, which measures mass, or nanomoles per liter, which measures particle number. These are not interchangeable. There is no universal conversion factor because the size of the apolipoprotein A isoform varies between individuals. The 2026 guideline uses 50 milligrams per deciliter or 125 nanomoles per liter as its threshold. The Canadian guidelines use 50 milligrams per deciliter or 100 nanomoles per liter, and the European guidelines have different cutoffs altogether. The FDA only approved a molar concentration method for LPA measurement in January of 2025. We are recommending universal screening in every American adult for a marker whose measurement method is not fully standardized and whose threshold for action is not globally agreed upon. Screen everyone with a test we're still calibrating for a condition we can't specifically treat yet. This is where we are. The 2026 LPA universal screening recommendation gets a Castelli coefficient of 5 out of 10. The science behind LPA and the causal cardiovascular risk factor is real and compelling. 30 years of prospective data, independent association with outcomes even after adjusting for LDL and inflammation. The biology is there. What's not there yet is a completed outcomes trial showing that lowering LPA specifically reduces cardiovascular events. A universally agreed threshold is also not there with a standard assay. And an answer to the question of what exactly to do differently for your patient just because their LPA is now at 60 milligrams per deciliter. A 5 here means the evidence for causality is solid. The evidence for what to do about it is pending. Maybe check back on this channel after Horizon and other trial reports are published. So there you have it. Can't exactly tell you what to do with the LPA recommendations. Just know that they're right now disease-oriented with no way to treat them. If you think it's going to help get your patients on lifestyle modification and make changes for the better to reduce cardiovascular risk, maybe it's worth it. Other than that, you might wait until you have more information about what LPA test to use and what you can do about it if it's high. Until the next time, this is Dispense is Written.